Good morning. Thank you for being here. I'm Ugo Bastolla from the Center for Molecular Biology, Severo Ochoa in Madrid. And my research focuses on developing integrated methods for analyzing protein dynamics, stability, and evolution through biophysical models, which is what I will tell you today. Specifically, I will tell about three tools that can be integrated in analyzing proteins, so classical protein bioinformatics. Start with PCALI, which is hybrid multiple protein alignment based on structure and sequence. We talk about TNM, torsional network model for predicting the statistical properties of protein dynamics in the native state. We'll finish with SSCPE, structure and stability constrained protein evolution model that builds site-specific amino acid profiles and substitution processes for inferring phylogenetic trees. So let's start with PCALI, which is a hybrid sequence structure alignment. As you all know, multiple alignments of proteins are very challenging, in particular for distantly related proteins. So it is convenient to use structural information. But structural information also reflects conformational changes besides homology. And moreover, there are two types of structural similarities. Those that require optimal structural superimposition, such as the TM score, which traces off alignment length and root mean square deviation, and those that do not, such as the contact overlap. So these similarity measures are correlated, of course, but they are both important. We cannot dismiss any of them. Therefore, we introduced the hybrid similarity measure PCSIM that integrates sequence similarity, spatial superimposition, contact overlap, and alignment length, just combining them as the principal component of these four correlated similarity measures. And we developed the PCALI, which is a multiple alignment that iteratively optimizes PCSIM. So PCALI is an iterative algorithm that needs a starting alignment for computing the matrix of structural scores, starts aligning all sequences, and classes structures of the same protein groups of conformations. This is particularly important when we are analyzing Cas superfamily, because we discovered that many files with Cas superfamily contain identical sequences, even if they are supposed not to do so. Then PCALI modifies the starting sequence alignment by maximizing the TM score, modifies the resulting alignment by maximizing the contact overlap, and finally modifies the resulting alignment by maximizing the integrated similarity measure PCSIM, and it obtains all pairwise alignments, and it builds with them a guide tree based on the divergence of PCSIM. It aligns proteins along the guide tree by maximizing PCSIM, and now it obtains a multiple alignment. And it uses the new computed PCDiv for computing a new guide tree and iterates the previous point until the similarity measure PCSIM does not increase anymore. So this will be the resulting alignment. As input, you can give the program either a pre-existing multiple sequence alignment, and then the program will output you all the structural similarity measures for this alignment, or a list of PDB codes, optionally with their domain decomposition CAS format. So it can be really applied to CAS superfamilies, and the typical execution time is some 60 minutes for superfamilies with 200 node redundant structures. So it's not very fast, but it's affordable, definitely. And the quality that it provides is good. So the resulting alignments are very similar to the manual reference Bali base, and they have a strong blockwise structure. They provide higher sequence similarity than purely structural alignments, such as TM align for Mammoth, and higher structural similarity than purely sequence aligners, such as NAFT, Kickoff, Cluster, or Moscow. So since PCALI outputs pairwise similarity and divergence scores, we can analyze, as we did in the upper figure, we can analyze the differences between the structural scores and the sequence scores provided by PCALI and the other aligners. So when it's positive, it means that the PCALI provides higher similarity measures, and as you can see that the alignment length is always higher for PCALI, almost always. the structural scores are similar or higher than for structural alignments, which are Mammoth and TM align and Bali based, and they are much higher than for the sequence alignment. And the integrated PC score, of course, is higher for PCALI, and the similarity with Bali base is also higher. So the quality is comprehensively better than for any other individual aligner, and integrated divergent measure PCD correlates very strongly with other divergence measures based only on sequence or on structure, so it is expected to estimate better the divergence time and to produce more accurate phylogenetic trees. And indeed, PCALI will provide us output phylogenetic trees based on PCD that we can use to detect accelerations of protein structure evolution, because we can measure the branches in units of sequence divergence or structure divergence. So we can see when there is an acceleration of structure divergence with respect to sequence divergence that we recognized as a hint of functional change in our analysis, the molecular clock of protein structure. Okay, so you can download the tool from the GitHub repository, where you also can find some documentation in form of README files. And it's very easy to install a new system. Just you have to unzip the package and run make. And you can run with command line options, where you have to specify, for instance, a list of PDB files with the option PDB list. And we have to specify the folder where the PDB files are stored. And if with the option print PDB, we will also get the structural superimposition PDB format. So the input can be, the PDB list input can be in CAS format. It's only mandatory to provide the PDB identifier. Then you can optionally provide the second column with the chain, the third column with the domain limits, with the domain name, and the folder where the PDB file is stored, because you can have two different folders. For instance, one for storing experimental PDB files, and another for storing AI predicted structures. And as output, you will get the multiple sequence alignment of secondary structure, summary statistics, statistics of conservation, and the 4 phylogenetic trees with the average linkage or the neighbor joining method, where the branches are defined as sequence divergence or structural divergence. So if you run the program without any argument, you will get help with the options, different options for providing, for instance, the data as a multiple sequence alignment, and the different output options for printing the structural superimposition, for printing the matrix of structural similarities, or for printing the matrix of structural divergences. Okay, that's all for PCALI. And now I'm presenting the torsional network model for native protein dynamics, TNM. It belongs to the class of elastic network models that predict the thermal dynamics of proteins based on their static structure, assuming that fluctuations are small, which is called harmonic approximation. It is the main approximation of physics, and that the force field is derived from structure, which in the protein literature is called the GO model. Under these assumptions, the elastic network models provide analytic results equivalent to averages of extremely long trajectories, so you don't have to bother about convergence. They're computationally fast and robust. In our case, there are only 3 global parameters that we optimized by comparing predicted and observed fluctuations in NMR ensembles. And in our model, the kinetic energy considers all atoms, and the interactions also consider all atoms, just choosing the closest atoms of each residue pairs, which is an important screening. redundant interactions. And you can choose the degrees of freedom. They are rigid body and torsion angles, but you can choose which torsion angles you consider movable. Only the phi angle, phi and psi, all backbone angles, phi, psi, and omega, and all backbone angles and side chains. Okay, sometimes you get sufficiently good results only with one torsion angle, which speeds up the computation and makes also the program more robust. So you also download it from the GitHub repository, github.com Ugo Bastolla. And you can install as the previous package, just unzipping a package and running make. And if you run the program without any argument, it will give you a list of command line options. You can also provide the input through a configuration file, which is provided in the package. So the main outputs that I will shortly tell you now are normal modes in torsional and Cartesian space, predicted Cartesian and torsional fluctuations, predicted dynamical couplings between pairs of residues and between and within functional sites, and analysis of conformational changes, possible functional role, transition paths, and finally prediction of structural effects of mutations. So first, you obtain normal modes, which are independent collective coordinates of the protein that perform harmonic fluctuations. Okay. With the option print PDB and specifying the number of modes, for instance, 6 modes, it will print 6 modes in PDB format, these PDB files, and then it will print the same modes also as Cartesian coordinates, the modescart file, as torsional coordinates, as modes tors that for all the torsion angles, and this mass-weighted torsional coordinates in MW-tors file. The file modes.dat will report the properties of all modes, so the frequency, collectivity, contribution to fluctuations. You see an example here. They are ranked by the inverse of the frequency, which is proportional to the contribution of the motions to the of the mode to the thermal dynamics of the protein. And important is the collectivity, so the fraction of atoms that it moves or the fraction of torsion angles, weighted or not, with the mass that it moves. So high collectivity modes are the important ones because they move important part of the proteins, such as interdomain motion. Then we get as input the predicted Cartesian and torsional fluctuations. In order to predict these mean square fluctuations in angstrom squared, we need to fit the scale of the predicted motions. And the TNM program does this by fitting the B factors of the experiment with the internal motions that it predicts. This is just one fitted parameter, the one that we are interested in, which is the force constant of the model. And then the rigid body motions in the crystal cell, and this is very important because rigid body motions represent most part of the B factors. These are 9 fitted parameters. Therefore, we have to regularize the fit. Otherwise, the fitted internal fluctuations may be negative, even if we disregard for rigid body motions. We only consider, for instance, one parameter, the translation of rigid body motions as it is done normally. We can get problems of negative force constant. So we use the methods of rescaled rigid regression that we proposed in this context with optimal regularization parameter. And it's very important to regularize. Every time you fit, you should never dismiss regularization. Otherwise, you can obtain completely unphysical results that are due to overfitting. So we found that only 20–30% of the B factors are indeed due to internal fluctuations, and the rest is rigid body motion. We cannot be dismissed. But normal mode analysis also allows to analytically compute dynamical couplings. And the classical one is the covariance, just the scalar product of the fluctuations of residue I and J. but we also define the codirectionality, which is the scalar product of the direction of the fluctuations, the residues I and j, which is more precise, for precise motion is more important. The coordination, which is the fluctuations of the distance between residue I and j, which is important for residues that bind the same ligand. And the deformation coupling, which is the deformation induced on residue I by force exerted residue j. So we found that the dynamical couplings change in a concerted manner of reaction cycles such as the leucine transport across the membrane by the neurotransmitter GUT, which is shown in these graphs. Every point represents a conformations along the reaction path, and you see this in the occluded conformation in which the leucine is inside the protein, in the interior of the protein. There is a highest coupling, and in the inward conformation, when the leucine is released to the cytoplasm, all the couplings decrease. So the analysis of the couplings can get you some insight on the molecular mechanism of action. You get the couplings with the options coupling, and you get files with the couplings between residues. It will print only the strongest pairs to avoid to get very huge files. And in the file which is called the profiles.dat, you get the mean coupling across all residues. So typically functional residues have a large mean coupling with all the rest of the protein. And then you get also the couplings focused to the binding sites. The binding sites.dat file reports the coupling within binding sites, so between the residues that belong to the same binding sites. The pair sites file reports the coupling between binding sites, so between residues that belong to different binding sites, and you see in this graph that this gives useful information. You have to provide information on the binding site, which usually it was contained in the site record of the protein data bank. So the program automatically reads this site record, but unfortunately, the site record recently disappeared from PDB files, and we must compute it internally and provide it with a file which is just the site of the PDB file, the site record of the PDB site. Then we can analyze conformational changes in torsion angle space. For doing this, we project a conformational change on the normal modes to the regularized fit. Again, it's very important to regularize the fit of the changes of torsion angles that better describe the Cartesian displacement. It's not enough just to compute the difference between torsional coordinates, because we are completely neglecting the other angles. So and so we would get a completely wrong result just considering the difference in torsion angles. So according to linear response theory, the contribution of a mode to the conformational change increases with the inverse of its frequency squared. We adopt a null model in which these two quantities are proportional, and we assess whether the low frequency modes contribute to the conformational change more than expected based on the null model. In this case, the harmonic energy barriers are lower than expected based on the null model, and motion is likely to be important for protein function. So you want to have this conformational change frequently in the intrinsic motion of the protein. And the parameters that tell you this are reported in the summary.dat file. So it's important the ratio between the harmonic energy barrier for the null model divided by the real energy barrier. If this is large, it means that the real energy barrier is much lower than expected. And it's important the correlation between the contribution of normal modes to the conformational change and the inverse of the frequency square, so its contribution to the thermal motions. This usually is very large, and the excess correlation. This tells when this excess correlation is significant, this tells you that the low energy modes contribute more than expected by chance. So this motion is likely to be functional. Indeed, it is the opening and closing motion of the transporter protein. Finally, we can use the TNM for predicting the structural effects of mutations. Again, we adopt linear response theory. We adopt a model by Julian Echave, which represents mutations as perturbations and predicts the deformation of the protein with linear response theory expressed through normal modes. So to finally specify, fully specify the model, we have to model mutation, and we do it through the change of the optimal distance and the stability of the mutated amino acid with all its contacts, and this change of size. Then the program predicts the global root mean square deviation and the destabilization of the native structure, which we call DE, caused by the mutation. And if you specify the option pred mood, the program will print files mut.de and mut.rmsd with the predicted effects of mutation on DE and the root mean square deviation for all possible 19L mutations at every site of the protein. Okay. And it will also give you a mutation profile file that tells you which is the local effect of any mutation on every site of the protein. So you will see that if you perform the mutation sites that have many contacts, you will get a higher response, a higher root mean square deviation, and higher perturbation. But the sites with many native contacts are less modified by the mutation. This is important for evolution, which is the last step where we developed the substitution models of protein evolution with selection on protein fold instability and protein structure, the SSCPE models. Okay. The empirical substitution models used for phylogenetic inference ignore site specificity due to the protein structure, although it is known that this affects evolutionary rates very strongly. So we developed the SSCPE substitution processes with fitness based on predicted structure changes and fold stability changes. These models only fit one parameter per protein, which represents the strength of natural selection, so they avoid overfitting, and they predict more realistic sequence variability, substitution rates, and hydrophobicity that no site-specific models, so empirical substitution models, or models that constrain only stability or only structure. And they infer phylogenetic trees more similar to structure-based trees. So, okay, the first ingredient that we have to predict is the stationary profile, PAOI, which is the product of a global amino acid frequencies, which are due to mutation process or selection at the level of the wood protein, and the site-specific logarithmic fitness for folding stability and structure maintenance that we compute from our models. We don't fit them. And the only fitted parameters is lambda, the strength of natural selection. So we have two stability constrained models. The wild-type model estimates fitness as the folding stability delta G of the mutants of the wild-type sequence using our method delta G rhyme that considers unfolded and misfolded states, also wrongly folded states. The Millfield model estimates the fitness self-consistently as the mean of the folding stability averaged over this. Then we have two structure-constrained models, and the RMSD that estimates the fitness as minus the RMSD of the mutation predicted with the TNM program. DE estimates the fitness as minus the destabilization of the native structure DE predicted with the TNM program. And then we have 4 structural and stability constraint models that combine the stability constrained and the structural constrained models. So then we need to compute the site-specific substitution models that tells you the probability that amino acid B substitutes amino acid A at a site I. And for this, we need the rate matrix QI, okay, which is standard in molecular evolution, is obtained as the product between the stationary profiles due to natural selection and the exchangeability matrix. So for each profile, we adopt 3 different types of exchangeability matrix. First, we adopt the Alpern-Bruno model, and so the site-specific exchangeability matrix is the product of a global exchangeability matrix, like from the empirical models of molecular evolution, times a site-specific factor that reproduces the Kimura's formula for fixation probability as a function of fitness. The no Alpern-Bruno version, the exchangeability matrix is not site-specific, it is global. And we have the flux flavor, in which the mean flux of each pair of amino acids equals the flux of the empirical model, and the no flux version, in which the global exchangeability of each amino acid pair equals the exchangeability of the empirical model. So we just use the empirical exchangeability matrix of the empirical substitution models. The phylogenetic programs that we use, such as RAXML and G, internally normalize the rate. So this is the option rate 0, where the site-specific rate disappears. And this just implemented because something which is done by the programs that we use. But we can also tell the programs to use the rate to input, and this is the option rate 1, where the substitution rate at each site I is the same as in the SSCPE model. So the best results are usually obtained with the options Alpern-Bruno 1, Flux 1, and Rate 1, but this depends on the particular data sets, and we can use all possible versions of the exchangeability matrix. So this is the flux of the program. Given one or more representative protein structures and an optional multiple sequence alignment, the program ProTable predicts the fitness of each amino acid at each protein site based on the predicted effect of mutations that we compute with delta G-rem, and the predicted effect of mutation on protein structure conservation, which we predict with the TNM program. Then ProTable fits the selection parameter lambda to maximize the likelihood of the multiple sequence alignment or of the input PDB sequences. And it derives 8 amino acid profiles, one for each possible models, the wild type, field, and the square deviation models, and so on, at each protein site, each with its fits and fashion, and each with its fitted value of lambda. They constitute the stationary frequencies of the amino acid substitution model. For each profile, ProTable estimates the best exchangeability matrix for various choices of Alpern-Bruno, Flux, and Rate models, and various empirical matrices like the Jones, Thornton, and Taylor matrix, which is very used in molecular evolution studies, the William and Goldman, and the Lee-Pasquale empirical exchangeability matrix. So the program SSPE.pl is a master program that runs ProTable or reads its outputs, its site-specific files, if they already exist in the folder where you run it. And for the chosen profile, it computes the exchangeability matrices, obtaining a substitution model at each site for the optimal model which is determined by Protable or for the choice required by the user. Finally, SSCPE.pl runs the program RAXML-ng, inputting to it the prescribed site-specific substitution processes. For example, the EMF, where the structure conservation is the E, and the stability conservation is mean field, and the Alper-Bruno, we use the Alper-Bruno model, use the flux model, and we use this prescribed rate. There are in total 8 times 8 times 3 choices of possible SSCPE models. With this model, it infers the sequence-based structure-aware phylogenetic tree with the program RAXML-ng. So also in this case, you can download it from the github.com Ugo Bastolla repository. But to install, you have to run in installation programs, install SSCPE.pl, which compiles and installs in the proper folders both the TNM program and the Protable program. You should also install RAXML-ng, but you may also find one copy in the package, although it is recommended that you install from the proper installation style of RAXML-ng. So if you run Protable without any option, you will get the list of options, input and output. You can write Protable with single PDB file, or you can run it with multiple PDB files, so a PDB list file, as those that uses PCALI. And in this case, you have also to specify the folder where the PDB files are stored, and you will get as input the site-specific amino acid profiles of the various flavors, and also the site-specific rates and so the information on the average properties of each site according to the model. So the rate, the sequence entropy, the hydrophobicity, and etc., etc., which will be interesting for downstream analysis. You will get a summary on the properties of each SSCPE model. So the improvement with respect to the global mutational model that does not consider sequence site specificity. And you will get the best model that maximizes the likelihood or an approximate likelihood of the multiple sequence alignment that you provided to the program. SSCPE.pl, also this program, if you run without any argument, will provide you a list of possible options, and you can run multiple PDB and infer the trees with RAXML-ng just with the option -RAXML, and you can either choose the model or you choose the default is the optimal model is reported by Protable. So finally, the perspectives is to integrate the profiles of SSCPE in PCALI in order to obtain site-specific and structure-aware sequence similarity score, and to develop new tree inference tools that regularize the maximum likelihood of least square fits of branch lengths, which is a crucial fit in the phylogenetic inference, but it produces overfitting because the current phylogenetic methods do not regularize these fits. So we are developing the Reckman score. So this is all. I hope that I was in time. I thank you very much for the attention, and these are some of the persons that participated in some stages of this work, and these are the funders. Thank you very much, and now I would like to take questions.